Good afternoon to everybody, and welcome to the third seminar of Bridges, which are seminars organized by the Hellenic Initiative Against Alzheimer's Disease, and their aim is to facilitate the dialogue between clinicians and basic scientists working in the field of Alzheimer's disease. So it's a honor for Bridges today to host as speakers two renowned scientists working in the field of mitophagy. They will talk about mitochondria, mitophagy, and neuroprotection applications in treating Alzheimer's disease and beyond. Our two speakers are Konstantinos Palikaras and Evandro Fei Fang. Konstantinos is Assistant Professor of Experimental Physiology at the Medical School of the National and Kapodistrian University of Athens in Athens, Greece. His main interests include the molecular mechanisms of autophagy, mitophagy, and their interplay between cellular metabolism and aging, and the development of novel genetic tools for biomedical research. For his scientific accomplishments, Konstantinos has received several notable scientific prizes, including the ERC Starting Grant in the year 2022. Evandro Fei Fang is an associate professor. Our second speaker is an associate professor from the University of Oslo in Norway, with his laboratory working in the field of the molecular mechanisms of aging and age-predisposed Alzheimer's disease. Evandro has received many awards, including the Norwegian Health Association's Dementia Award, presented by His Majesty the King Harald V of Norway in 2023. So, Konstantinos, the floor is yours.
So, first of all, I would like to thank you for the opportunity. I would like to thank Dr. Akershus and Dr. Bachiraki, Ioannis, Panos for also accepting me in this HIAAD initiative. I'm really happy about that opportunity. I'm really thrilled to work with you and collaborate with you to push the boundaries of our research and try to identify the molecular mechanism of neurodegeneration in Alzheimer's and maybe help the patients in future. So, let me share my screen. Can you see my screen?
Yes, perfect.
Yes, perfectly.
So today I will talk about the neuroprotective role of mitophagy in Alzheimer's disease. And so probably most of you already know that neurons contain increased mitochondrial number to carry out their functions and preserve their homeostasis. And also neuronal cells have a unique cellular architecture which is characterized by a relatively small cell body, long axons, and multiple dendritic branches. Thus, neurons have developed specialized molecular mechanisms for efficient distribution of mitochondria to the most distal parts of the neurons, such as growth cones, post and presynaptic headings, where high energy is required to sustain their activity. So post-mitotic neuronal cells must survive during the lifetime of the organism, and they are more vulnerable to the process of aging due to their intensive metabolism and increased growth generation. So the neuronal activity and survival depends on proper mitochondrial homeostasis. So dysfunctional mitochondria not only generate less ATP levels, but also display impaired cytoplasmic calcium buffering, increased release of apoptotic factors, and elevated growth generation, which subsequently lead to neuronal stress and neurodegeneration, and may lead also to premature aging, autoimmune diseases, or even neurodegeneration. So several mitochondrial quality control mechanisms have been developed during the evolution to sustain mitochondrial homeostasis, such as mitochondrial dynamics, which are very important through fusion-fission event to dilute mitochondrial damage in mitochondrial population, the proteasome system, mitochondrial-derived vesicles, and mitochondrial alpha-folding protein response. When these pathways cannot repair mitochondrial damage, then this damaged mitochondria needs to be eliminated through autophagy, through a process known as mitophagy. Neuronal mitophagy is a challenging event since the majority of mitochondrial mass, as I already stated, it's located at very distal parts of the neurons and need to travel back to the soma where mature lysosomes are mainly exist. So although this spatial limitation, rapid mitochondrial elimination is really important for the maintenance of neuronal activity or even the protection against cell death. So during aging, all these mechanisms are deteriorated, leading to energy imbalance, ionostasis, impairment, synaptic failure, and cognitive decline. So damaged mitochondria are characterized by increased mitochondrial DNA mutations, increased ROS production, reduced membrane potential, and misfolded proteins. So these damaged mitochondria needs to be targeted specifically and eliminated through mitophagy. So during the last decade, several molecular mechanisms of mitophagy have been described. The most well-known is the PINK1 parking pathway. So when we have damaged mitochondria with impaired membrane potential, PINK1, protein kinase, mitochondrial protein kinase that is localized on mitochondria and upon mitochondrial damage is stabilized on the outer mitochondrial membrane and start the phosphorylation of several molecules, including ubiquitin and also parkin. Parkin is an E3 ubiquitin ligase, and upon its phosphorylation is recruited on damaged mitochondria and start to ubiquitinate several outer mitochondrial membrane protein. So upon the generation of polyubiquitin chains, these damaged mitochondria are recognized by the autophagosomal proteins, LC3 or adaptor molecules, and these damaged mitochondria are specifically engulfed and delivered to lysosomes for degradation. Of course, there are other pathways that are PINK1 and parkin independent. So we have the receptor-mediated mitophagy. So there are proteins that are localized on the outer mitochondrial membrane, mitochondrial proteins, and serve as mitophagy receptors. So these proteins directly interact with autophagosomal protein LC3, and upon their interaction, these mitochondria are engulfed and delivered to lysosome for degradation. Additionally, the pathways became more complex, and there is an intricate communication between the PINK1 parking pathway and the receptor-mediated mitophagy pathway. So they are not two pathways that cannot interact at all. So this it is already known, and a growing body of evidence suggesting that the PINK1 parking pathway phosphorylates or ubiquitinates several mitochondrial receptor, mitophagy receptor, and upon these post-translational modifications, receptors are activated, and now they are ready to interact with the autophagosomal machinery. So to highlight the significance of mitophagy process, the majority of mitophagy players are evolutionarily conserved across taxa, from yeast to humans, signifying the pivotal role of mitophagy in organismal physiology. Additionally, mitophagy and autophagy efficiency declines with age, and this leads to increased prevalence of age-related diseases, including neurodegeneration. So we can say that autophagy and mitophagy dysfunction is a common denominator of age-related diseases and neurodegeneration. So the central aim of our group is to understand the molecular mechanism of aging and how aging affects the nervous system, focusing on mitochondria, because mitochondria provides 90% of the energy that is required for brain development and function. And of course, we're focusing on mitochondrial quality control mechanisms to see how this mitochondria, the mitochondrial homeostasis is preserved during aging or neurodegeneration. So we use C. elegans as a model organism. C. elegans is a soil-dwelling, non-parasitic nematode. It is transparent, so this makes an ideal model for microscopy research. C. elegans has an exceptionally well-characterized anatomy and development, has a fully mapped neuronal network. We know the entire connectome of C. elegans. C. elegans has 302 neurons. We know the complete wire diagram. We know the number and the nature of the synapses, their spatial and anatomical position. And there are some features that facilitate C. elegans as a perfect model organism for aging research. C. elegans has short lifespan, only 2 weeks is the median lifespan, and 30 days is the maximum lifespan. And finally, C. elegans is a hermaphrodite worm. So upon self-fertilization can give rise up to 300 genetically identical progeny clones. So when we are performing lifespan assays on any other experiment, we have reduced genetic background mutations, or genetic backgrounds are very similar. So we don't have huge variations in our experiments. So this is how C. elegans look like under the stereoscope. You can see nematodes here and different larva stages and embryos, eggs. And C. elegans eat E. coli, an E. coli strain called OP50 that is not pathogenic. So we use this organism to monitor lifespan assay upon different stimuli, different treatments. We can monitor toxicity during development and their reproduction rate. And of course, we can monitor locomotion, their motility upon overexpression of pathogenic proteins or upon the treatment of specific substances very easily by using just the camera and a stereoscope. So the goal of our research is to investigate the molecular mechanism of mitophagy and how mitophagy affects the aging process. Of course, we are performing drug screenings in collaboration with the Department of Pharmacy here in Athens to identify naturally occurring compounds or synthetic compounds that promote mitophagy and affects physiology and lifespan. We like to develop novel nematodes models for human neurodegenerative diseases. So we are in close collaboration with the Genedis Hospital and Professor Stefanis that give us new mutations to generate that have been identified in human population, and we try to generate new models of Parkinson or even Alzheimer disease. And finally, we like to develop and optimize novel genetic tools for biomedical research. So we are members in aging societies. The world population is aging every day. So many countries, especially those in the European Union, either have become or are currently becoming aging societies. And there are estimations that says that the aging population will be double during the next decades. And as people age, the risk of developing neurodegenerative pathologies increases, and neurodegenerative diseases such as Alzheimer, Parkinson, or ALS. So most of these diseases have modeled in C. elegans. So we have modeled for several neurodegenerative disease models. And of course, we have modeled for Alzheimer that we use in our research. In fact, Alzheimer is the most common dementia disease and neurodegenerative disease in human population, in aging human population. The impact of Alzheimer's disease in aging population is significant, both in terms of individual health outcomes, but also the broader aspect in societal impact. And it's really shocked that over 200 years of research, and still we don't have any therapeutic, efficient therapeutic approach intervention for the disease. So we focus our research on Alzheimer's disease, and we use several AD nematode models that exist in C. elegans. We use a humanized model that overexpress the A-beta peptide or the tau protein paneuronally or in other tissues such as the body wall muscle cells. And then we try to investigate the molecular mechanism of neurodegeneration and also to identify novel drugs and therapies. So a recent study from our group, when me and Evandro were still postdoc in the lab of Professor Wilhelm Bohr and Nektarios lab in Stavranakis in Crete, we identified that mitochondrial dysfunction and defective mitophagy are hallmarks of AD pathophysiology. So here we can see AD post-mortem samples, and we identify increased mitochondrial damage. We also find out reduced mitophagy levels in human post-mortem samples. We identify similar results in AD nematode models. We find out increased mitochondrial damage, reduced basal and stress-induced mitophagy, and also we found that nematode AD models display cognitive defects. So we decided to perform screenings and identify novel mitophagy modulators in order to reduce damaged mitochondria and to see whether we can promote neuroprotection. So in order to do that, we develop in vivo imaging systems to follow up mitophagy. So this is what we did. We generated a stable transgenic HILA cell line that express the mitochondria. Mitochondria is a fluorophore that change color from green to red depending on the pH. So when the pH is acidic, inside the lysosomes, upon diffusion with lysosomes, mitochondria are red. So by measuring the GFP and the green-red ratio, we can signify mitophagy events. And also we generate transgenic nematodes expressing in paneuronally another biosensor called Rosella, which is a biosensor that's comprised of a pH-sensitive GFP, fuse the pH-insensitive DS-red. And we send this biosensor on mitochondria, and upon mitochondria engulfment by autophagosomes and fusion with lysosome, the GFP signal quenched, however, the DS-red signal remains stable. So we perform drug screening and we identify 3 potent mitophagy inducers: urolithin A, a natural occurring compound coming from pomegranate seeds and some other red berries that trigger mitophagy and increase lifespan in worms and muscle function in rodents. We identify nicotinamide riboside, NR. It's an AD precursor molecule that is known to promote energy homeostasis and lifespan. And actinonin, it's naturally occurring antibacterial agent. So all these drugs promote mitophagy. So we treat the AD nematode model with these molecules, and then we test their associative learning capabilities. And we find out that both the tau nematode model and the A-beta expressing nematodes, upon their supplementation with these 3 compounds, we observe associative learning improvement. So urolithin A, nicotinamide riboside, and actinonin supplementation restore cognitive function in AD nematode models. And then we try to investigate the molecular mechanism, which through mitophagy mechanisms this is happening. And we found out that urolithin and actinonin requires PINK1 and parking to promote neuroprotection and associative and improve the learning capabilities, whereas NMN requires PINK1, parking, and mitophagy receptor BNP3. So here I'm summarizing these results, and I would like to emphasize that these neurons that regulate the learning and memory effects depends on dopamine. And why I'm stressing this out? Because then we monitor the motility effects, because these AD nematode models also display some motility defects. And we treat again these animals with mitophagy compounds, urolithin and NMN. We managed to improve the motility upon mitophagy upregulation. However, in this case, both NMN and UA requires all the components of mitophagy machinery, the PINK1, the parking, and the BNP3 receptor to promote neuroprotection and locomotion. So here, these neurons, the neurons that regulate locomotion in C. elegans, depends on GABA and acetylcholine, and GABAergic and cholinergic neurons. So what we can say now about this result is that mitophagy, this compound may induce mitophagy through different mechanisms in cell type specific manner. The neurons that depends on dopamine requires only PINK1 parking for urolithin upon urolithin treatment, and NMN requires PINK1 parking and BNP3. However, on the other hand, we have the locomotion and the GABAergic and cholinergic neurons that require all mitophagy components, the PINK1 parking pathway, and the mitophagy receptors in order to promote neuroprotection and improvement in motility. So then we checked more, and we took advantage of a very recent study that performed single-cell RNA analysis in C. elegans neurons, and we identified that mitophagy regulators are differentially expressed in neuronal cells. So our results can be explained by this data and make our story even more complicated, and highlight that this is the case also for other neurons, that different subtypes of neurons regulate mitophagy through different mechanisms. So I hope that in the next years you will hear more about this work. and our findings. So let's now move on tauopathy model. In the tauopathy model, it's very well appreciated that tau aggregation affects mitochondrial homeostasis in AD models. However, what it is not known is whether this mitochondrial dysfunction is a largely pathological feature of high levels of tau, or whether this mitochondrial dysfunction is a consequence of excessive tau aggregates. So in order to address these questions, we took advantage a tauopathy model that exists in C. elegans and express at low levels the human wild type of tau protein. And it is already known that tau aggregates during aging, very late ages of adulthood, day 3, day 6, and day 9 nematodes. However, it was not known what is happening earlier in earlier developmental stages. So we monitor tau aggregates, high molecular weight of tau at L4 and L1 larva stage, and we find out that tau aggregates is not taking place in L1 and L4 larvae, and we can detect only tau monomers. So then we monitor mitochondrial number, and we assess the morphology of mitochondria in actions of these tau models. And of course, we monitor the motility defects that these animals present. So here I'm summarizing the results of this study. We identified that tau aggregation is not taking place in L1 and L4 larvae, but only start to accumulate in day 3 and day 6, so during aging process. But we observe increased motility defects during the whole developmental stages and also larva stages and during aging. We observe low mitochondrial density at larva stages and high mitochondrial density later on in their age. So this can be explained by the fact that mitophagy is efficient, still efficient, very young animals. However, mitophagy efficiency declines with age. So during aging, we have accumulation of human tau, but also increased mitophagy deficiency that leads to increased mitochondrial accumulation. We observe during all developmental stages globular mitochondria and small mitochondria that highlights that we have increased mitochondrial damage that is accompanied by decrease of mitochondrial membrane potential. So all in all, what we conclude is that tau expression is sufficient to promote mitochondrial dysfunction and impair cellular function. And during aging, tau aggregation even make the story worse, and we have reduced health span, excessive mitochondrial defects, and neurodegeneration later on in their age. So now we use these animal models, mitophagy reporters, mitochondrial reporters, and these tauopathy models. We are in agreement with the pharmacy school and Dr. Kartsunis that will give us 100 natural occurring compounds that are extract from Greek plants. And we will use a high-end microscope slide scanner that we have in the lab, combined with the formation tools, to monitor mitochondrial morphology, muscle function, neuronal function, and to identify novel mitophagy modulators. And then maybe we can propose or suggest some new drugs against AD pathophysiology. So in general, mitophagy inductions through NMN, nicotinamide riboside precursor molecules and AD molecules, or urolithin or actinonin, would promote mitophagy and promote neuroprotection through the elimination of several pathological features of AD, such as A-beta accumulation or tau accumulation. However, what we should keep in mind is that there is a balance inside the cell, and there is a coordination between mitophagy and mitochondrial biogenesis. And this balance needs to be maintained, because if we overdo it with mitophagy induction and we have excessive mitophagy, maybe we have the opposite effect, because there are evidence from other pathological conditions that we have increased mitophagy, excessive mitophagy, and reduced mitochondrial biogenesis, and that leads to decreased mitochondrial content and energetic crisis. So we have mitophagic cell death. Neurons die because of increased mitophagy. So always when we use treatments or interventions that promote mitophagy, we should also keep in mind that we should also promote mitochondrial biogenesis in order to keep the balance and the homeostasis. So I would like to thank now my group members, my postdoc, Christina Fibos, and my PhD students for their work in the lab, our collaborators, Wilhelm Bohr, Evandro Fei Fang, Lucas Korano, Nektarios Tavernakis, Leonidas Stefanis, and Paminodas Doxakis, our funding sources, and I would like to thank you for your attention.
Many thanks, Konstantinos, for this great talk. I think that many questions will be posed when the discussion part of the seminar will be opened. So, Evandro, the floor is yours.
Okay. You guys see my slides?
Yes.
I hope you can see this video. So I think it was very nice. Konstas gave very detailed mechanistic understanding of Alzheimer's disease and mitophagy. So I will give a similar story, but in a more simple angle and a little bit more on the translational and clinical perspective to think how to bridge both the eyes from a basic researcher as well as half a clinician, because I got my training bachelor and master both on medicine. What I showed you just now is the grandma, Betty, who is 85. And you can see she is even unable to recognize herself in the mirror. And this is because of damage of her memory. And for her case, it is Alzheimer's disease. I want to give a brief introduction for myself. So I got my PhD in Hong Kong. Then I went to Baltimore for aging research. Many of people ask me why I moved from Baltimore to Norway. I think one of the reasons was Wilhelm Bohr, as a Danish American professor who shared with us a lot culture of the Nordic countries, and have been maximized by the research standard and environment in Nordic countries. For example, his father, Niels Bohr, who got the Nobel Prize in Physics in 1921, and was visited by Queen Elizabeth, her husband, as well as the Danish Queen and the King, which highlights the highly respect to the scientists. In my group, there are three major topics. The first one is the molecular mechanism of aging. We focus on a small molecule called NAD+, which reduced dramatically when we are getting older, as well as in Alzheimer's disease. We are interested in why NAD is reduced during aging and how we can boost NAD levels to maintain healthy longevity. And my second topic is Alzheimer's disease. With Konstas as our long-term collaborator, I have been interested in the garbage accumulation as a cause of Alzheimer's disease. And we are interested in increase the garbage recycling capacity as a novel therapy for Alzheimer's disease. And we have been using artificial intelligence as an approach to prepare drug development targeting on aging and Alzheimer's disease. And today I will just touch base on different subtopics. In addition to to C. elegans, as Konstantinos mentioned, there are many model systems used in the aging field. While my group uses C. elegans, Drosophila mice, as well as human postmortem brain tissues for aging and Alzheimer's disease research. If you look at the picture of Betty, when she was getting married at her 20s, she had a perfect memory. She only had memory impairment when she was at her 70s and 80s, which indicates aging is the primary driver to cause the disease. If you look over details regarding people's brain, this is a schematic model. When we are young, we have very clear and healthy neurons. However, for patients died of Alzheimer's disease, when we cut their brain and we see accumulation of those garbage, including extracellular A-beta plaques, intracellular A-beta peptide, as well as intracellular tau tangles. And we believe accumulation of those garbage are causes of the neuronal dysfunction. If we take it in a simple way, think about the city, Paris. This is the picture in our mind of Paris. However, you may already saw from the news and TV, because of the strike, and Paris has been packed with the garbage, which blocked the running of the city. And the common way for garbage recycling, it is to having the truck. And as Konstantinos mentioned, mitochondria plays an important role in having this garbage trunk to move. And our interest is when we are getting older, the worn out of the garbage of the engine, which reduces the recycling capacity of the garbage truck and causes accumulation of the garbage. And we aim to fix the damaged engine. What we can do? There are many approaches. For example, exercise, fasting, medicine. There are some medicine available which can alleviate some of the syndromes. And yesterday, there was an anti-A-beta drug in stage clinical trial showing it was able to reduce 35% of Alzheimer's disease development. But the side effects, it has a huge side effects to 25% of the participants with brain shrinkage and blooding. In addition, diet, a healthy diet also plays a role. In addition to those four, one parameter which has been greatly neglected but plays an important role, that is mental health. So I will touch base on some of those topics in the following 10 to 15 minutes. Dr. Shu Jingcao, a postdoc in my lab, she did her PhD in Thailand, a country that consumes a lot of colorful fruits, especially passion fruit. And there is epidemiological evidence showing patients having colorful fruits, including passion fruits, has a likelihood of better quality of health, as well as reduced chance of dementia. Since those epidemiological studies cannot pinpoint whether it is a causative effect or it is a bystander, so Shu brought this question and joined my group. And she wanted to know whether passion fruit has any molecules inside can really inhibit memory loss in animal models. And the answer was yes. And she has identified 2 molecules which were able to induce mitophagy in the neurons in the brain of the mice to reduce human iPSC-derived neurons, astrocytes, and microglia combined co-culture system. And 2 weeks ago, we had very exciting results showing EFFA molecule was able to pass the blood-brain barrier, which further consolidate its potential translational application. And the paper is in preparation. Before Shu was writing this one, she had another paper showing the crude extract isolation from passion fruit was able to reduce memory loss in animal models, and which we got the news from the Norwegian national channel. And believe or not, the second day when we went to the market, and all the other fruits are there, but the passion fruits was gone. So it really means the people really following the scientists' work. That gives us more encouragement to dig into more details and to be more responsible on our work. The second topic I want to share with you is NAD+. As Constance mentioned to you, this is a molecule can induce mitophagy. However, this molecule is dramatically reduced when we are getting older. For example, people age 65, there's an average 75% reduction of blood NAD compared to that of your 20s. And by the application of NAD and other molecules as mitophagy inducer, in addition to increase memory, as Constance mentioned to you, I want to share with you the validated data in mice. This is a AD mouse model, and you can see with the mitophagy drug treatment in the right side, the mouse were more targeted on a region close with the platform, and it was able to target to find this platform in a very short time, while the litamate without any drug treatment had a harder time to find the target, which indicating the drug can potentially increase spatial memory and learning. So if we give a summary of the data Constance mentioned to you in C. elegans, and some data I mentioned as well as not mentioned in human brain, human iPSC, and mice. Our data consistently enabled us to propose a hypothesis that during aging, genetic and non-genetic factors that can increase defective mitophagy, which drives accumulation of damaged mitochondria. We would like to point out that this process may not be sufficient to cause Alzheimer's disease. We still need risk factors of Alzheimer's disease, and tau, A-beta, they are intertwined with impaired mitophagy to induce neuronal dysfunction, impaired engulfment of A-beta plaques by microglia, and this may contribute to brain shrinkage as well as loss of memory. Why we believe impaired mitophagy is at least a risk factor of Alzheimer's disease rather than a bystander, because when we genetically and pharmacologically reinstall mitophagy, for example, using NAD, uranase A actinoline, as Constance detailed in the worms, we saw increased mitochondrial quality, increased neuronal survival, increased engulfment of A-beta plaques, reduced inflammation, another risk factor of Alzheimer's disease, as well as reduced extracellular A-beta plaques, intracellular tau, intracellular A-beta peptide, as well as intracellular tau tangles, and which may finally contribute to better memory. And we are now using genetic approach to further investigate this thing. So here, I want to shift gear to go to the next one, which is exercise. And this guy and this guy, the top two, they are famous Norwegian cross-country skiers, and they got the world champion #1 and #2 a few months ago. Because exercise has been shown as a bona fide and very robust way to increase quality of life and reduce memory loss. And we wondered, in addition to exercise, if we put NAD+, can the combined therapy show better results? And this study is leading by DPhil student Helin Wang. So we forced the mice for 20 minutes treadmill exercise, and the results indeed show the mice had better memory than a single treatment. And this study is still ongoing. We are also interested in using artificial intelligence as an approach to prepare drug development. And as you may already know, AI has been used in almost all fields close to our daily life, including for medical research. So due to time concern, I want to give you a summary of this project we finished last year. We used AI and built an algorithm. And this is 3 types of machine learning approaches combined together, including vector representations of molecular structures, pharmacophore fingerprinting, and conformal fingerprinting. And then we had it trained in a large database, followed by getting access to the University of McCall-based small compound library, which contains over 3,000 natural compounds isolated from traditional Chinese medicine, which has written evidence of dementia benefit. And we were interested in which molecule may contribute to anti-dementia capacity. With the AI approach, if we give a score 1 as the best fits, we had a cutoff of 0.75, which enabled us to select the top 18 compounds. Then we went to the wet lab and see using this HIAAD-based mitochondria system at a 10 micromolar as a cutoff approach, and 8 compounds were able to induce mitophagy. And we first asked how many compounds were able to induce mitophagy in C. elegans, and 2 of them were able to induce neuronal mitophagy as well as to improve memory. Encouraged by those results, we then tested our results in a triple TG AD mouse model, harboring both tau and A beta pathologies. And luckily, after 2-month treatments from 11 months of age, those 2 compounds were able to improve, induce mitophagy, inhibit tau and A beta pathologies, and increase memory. I was very fortunate to be also affiliated to the Akershus University Hospital in Norway, which has the largest neurology department here, which enabled me to be involved in 5 NAD-based clinical. It has, I don't know, it's automatically go clinical trials, including on premature aging disorders, ALS, cardiovascular disease, as well as an Alzheimer's disease, phase 2 clinical trial with NAD treatment ongoing in University of Bergen in Norway. If you want to ask me any approach you can do in your daily life without taking a lot of effort to improve your healthy longevity, I would share with you that NAD+ actually it's available in both beer and milk. However, life is not that easy because it is very low traces in the beer. You need to drink a huge amount to reach the level of its biological feasibility. That is why many pharmaceutical companies are developing NAD precursor as diet supplements in the market. In addition, I want to share with you mental health is also important to take care of for dementia. I've been involved in this national documentary to show that actually a music choir can help to dramatically reduce memory impairment. This is because you have reduced the pressure and increased the social capacity, increased the brain activity. Doing Alzheimer's disease can't be done by one person. I was very fortunate to working with Professor Manuel Waiter. We had built this Norwegian National Anti-Alzheimer's Disease Network. Norway has put a lot of money on Alzheimer's disease research, and the former Minister of Health and Care, he said, we owe it to society to fund the cause of Alzheimer's disease. As I mentioned to you, Alzheimer's disease with aging as a primary driver. So it is important to consider the disease as a whole. We have been built this Norwegian Center on Healthy Aging Network, trying to using the knowledge from the aging researchers to help on the understanding of Alzheimer's disease. The final goal of us is to making us physically strong and mentally sharp at our 90s. And finally, I would like to thank my team members, my collaborators, my funding, as well as my team. Thank you for this opportunity.
Many thanks, Evandro, for this great talk. Very, very interesting results. And I suppose that what, in my opinion, it's really important and really interesting, your approach here, bridging work which is done in the lab with findings which from which patients and people living with dementia and older adults, and we all will benefit hopefully in the near future. So we do have time for questions. So please go ahead. Who would like to pose a question? Kostas?
Well, very impressive. I like the model. I like the process. I like the accumulated data. I have two questions. First question relates to the study of human brain tissue with Alzheimer's versus not Alzheimer's from autopsy material. Is there evidence that in that setting, there is accumulation of mitochondria specifically related to AD as opposed to age-matched controls? Last I looked, the data were somewhat controversial and that it wasn't clear. So I'd be curious to hear where that stands. And the second question, and this is something I think applies to anybody who's trying to develop new therapies in this space, which is safety ends up being one of the biggest concerns, as well as drug delivery and target engagement. So rather than look for new compounds, why not think of screening compounds that could be repurposed? And there are a number of libraries that are available through the NIH. At least you've got the safety, you've got the manufacturing capability, you don't need to worry about that. But if you have the screening paradigm, you know, from C. elegans to human IPS to human trial, you've got a ready drug that you can take into trial.
Thanks, Kostas Liktotis. I would like to answer the first one, and I will give Dr. Palikaras to respond on the second one. So the human brain samples we got from Harvard Brain Bank, we had 8 AD patients at their 60s to 70s, and we also has the sex and age match controls without cognitive impairment, but died because of car accident and different things. And in those small cohort, we indeed see accumulated damaged mitochondria in both the hippocampus region as well as prefrontal cortex region. And this is correlated with reduced mitophagy. And for mitophagy, we used a couple of methods, including using the classical electron microscopy to check mitotic evidence, as well as to use protein-protein co-localization method. Definitely, I also noticed there are inconsistency regarding how mitophagy, mitochondria autophagy changes. It could be because there are huge difference on humans. Even you have sex and age matched, there's still a lot of things can impact the results. For example, how many hours after the patient to be died and you collect the brain tissue, that can also matters. Nevertheless, we have now collected more than 100 Alzheimer's disease patients from birth stage 1, 2, 3, 4, and 5, 6 from King's College London. We had hippocampus, prefrontal cortex, as well as entorhinal cortex. So we are now doing more like a critical and comprehensive evaluation. And it seems the answer is still the same. And I give the second one to Dr. Palikaras.
Thanks, Evandro. Indeed, we are doing some new drug screenings, and we are set up some new drug screenings with compounds that have not get any FDA or EMA approval. So we try that because we have the collection of all these Greek traditional extracts in the Department of Pharmacy, but also we are doing drug reproposing. So we got a grant from a company called Helix, and they provide us with compounds FDA approved that get the FDA approval. So we are also performing screening with this kind of drugs that they have already passed the toxicity effects, and we don't see any toxicity. So we can use these drugs and get quick results, and we can reuse them in Alzheimer's disease or any other new AIDS-related disorder that we study. For example, in our case, we perform a drug screening with such kind of drugs for autosomal dominant optic atrophy. It's not an age-related disease, but it's a mitochondrial disease, and we perform drug screening for drug reproposing. So we are doing that too. But we like to identify novel drugs, some compounds from extract that exist in our country, and maybe we can use them as therapeutic agents in the future too.
Many thanks. Are there further questions? Ioannis Zaganas.
So thank you very much. Thank you very much for this very interesting talks. So about NAD, which I found it very interesting. Is it that you have a decrease in the during aging, a decrease in the total NAD, or is it a shift towards the NAD plus and not a decrease in the NADH, the reduced equivalent? And can this be, let's say, explained by reduced absorption of NAD from the gastrointestinal tract, different metabolism of NAD? What would be the explanation? Of course, I don't think that by aging you drink less beer. That would not be an explanation.
Maybe I can start first, then Palikaras, you can add some more.
Yeah.
So there has been a series of studies in mice, C. elegans, as well as tissues from humans like fibroblasts, as well as blood samples. I would say not all of the data, but most of the data show an age-dependent reduction. It's the rate matters. Some studies show a huge reduction, others show less reduction. And definitely checking NAD in the human blood, whether to using whole blood samples or PBMC or plasma or serum, has a debate there. And you are asking, because when we are talking about NAD, it contains NAD plus, the oxidized form, as well as NADH, the reduced form. In our mice, C. elegans, and human skin cell-based studies, in Alzheimer's disease, normal aging, as well as a series of premature aging diseases, we saw absolute level of NAD+ as well as the ratio of NAD+ to NADH are both reduced. Sometimes the ratio may not change, but the NAD+ absolute level is reduced. That was why we use NAD+ absolute level as a marker for us if there are any conflict between the two parameters. Palikaras, do you have anything to add?
I don't have something to add. I think that you covered the question. We know that during aging, NAD levels declines in several tissues, and it's true that's happening also in C. elegans, because we have the NAD biosensors and we have monitored NAD levels in different tissues in nematodes. This is something that we are planning also to do it again because we have some new imaging biosensors. I remember that you generated them, Evandro, recently. So, but in general, we know that the NAD levels decline during aging. So we try to boost them by using NAD supplementation exogenously. So yeah, I think all the others you covered.
So do you think it's a decreased absorption from the gastrointestinal tract or something else? Or do you think also it's about the ratio you said we can, let's say, shift the balance toward NAD+ and have less NADH, and this could be also beneficial?
So I'm sure that the gut absorption will participate in this problem in NAD deficiency during aging. But also during aging, we have increased DNA damage. And we know when you have accumulation of DNA damage, there are enzymes that are activated to repair the DNA damage. These enzymes needs and consume a lot of NAD. So the already low levels of NAD inside the cell are not enough for all the cellular processes. Also, that's why we should increase the levels exogenously. So one explanation is the decreased absorption from the gastrointestinal tract. The other is that also other processes that take place simultaneously during the aging process consume NAD. So that's why we need to boost it.
Many thanks. Are there further questions?
Panos, there is one from Konstantinos Palikaras. You can read it.
To check it. Another question from Konstantinos Palikaras is if you have studied SSRIs such as escitalopram with regard to mitophagy.
What is SSRI? Do you know?
SSRI, the selective serotonin reuptake inhibitors. Okay. One of the modern categories of antidepressants.
That should be very interesting. I think that could have some benefit. Yeah, it has been shown people, after your retirement without kids around you and depression alone, lonely, can at least increase the progression of Alzheimer's disease. There must be some clinical studies, I bet. But if you don't mind, I want to give one adding regarding the NAD-based clinical trials. My colleague in University of Bergen, they published a paper on phase 2A clinical trial for NAD+ precursor NR on Parkinson's disease. And using the MRI method, they show with the precursor supplementation, they saw increased brain NAD levels. Even this is a phase 1 and phase 2A project. They were able to see NAD+ supplementation was able to reduce not all, but some of the parameters related to inflammation in Parkinson's disease patients. And they are now doing a similar thing for Alzheimer's disease. So we are very excited. And I want to add to, because Likitos asked us why to do the traditional ones. I think one good thing to use NAD+ because this is a molecule available in our body. So you just supplement what we missed before. And there had been over 20 clinical trials showing any plus precursor is safe using the dose between 0.5 to 1 gram per day for up to 3 months. That is the data we have so far.
Many thanks. There are two further questions. Espen, please go ahead.
Hey, very good talks, very informative and easy to follow. I had a question about CD38 and senescence cells in general. If there's any interest in looking further at how this impacts NAD decline during aging. CD38 reduces levels of NMN and NAD, and it's caused by senescent cells. So yeah, I was just wondering if there's any interest in that in your lab groups.
Palikaras, maybe you can start first.
We haven't checked anything about senescence in that context because we don't have the term of senescence in C. elegans body. So we haven't used this kind of molecules for AD, at least in our lab in nematode. So no, I haven't, but it seems very interesting, and we know that senescent cells have very important function in during aging, and also that participation in the AD pathology. So yeah, I don't know something more than that. You know, Evandro, maybe.
Yeah, I would like to add a little more. So Alejandro, a Brazilian American colleague working in Mayo Clinic, he knocked down CD38 in the wild type mice, and he saw when the mice grow older, the NAD cellular and tissue levels were higher, and he also saw increased health span and lifespan in the animals. While I think those data are exciting, but I would like, so this is the number one. And number two, there was a study published by a Israeli group showing an Alzheimer's disease mouse model, I forgot which one, but with the crossing with the CD38 mutant mice, they saw increased memory. So this was in line with potentially reduced CD38, increased NAD levels, and increased memory. But I want to highlight that CD38 is also has its important roles, like yin and yang. It has been shown CD38 is important for social behavior. When you knock down this gene, the mice stopped to interact with each other. So we need to keep this in mind also. Thank you, Espen.
Thank you for the answers.
Many thanks. Nikki?
Yes. I want to congratulate both of you, Kostas and Evandro. It was really a delight to follow your talks. And I have a very, I might have missed about this NAD+. So what is the dosage? I mean, what are the evidence about used by humans? I mean, I know there are so many supplements in the market. What should somebody do? Do you have an idea? What is the dosage? I mean, I'm 64 years old, so I'm already in a very delicate age. I need to take care of myself. So what dose should I use?
Evandro, I leave this question to you because I'm sure that you know more, and especially I know that you used to get some NAD supplements in the past, so I'm sure that you will give a perfect answer to Nikki.
Yeah, so first I want to make a claim on the conflict of interest. I think there's only one company, because it holds a patent called Chrome Index, which can patent to synthesize this molecule. I do not have any, I do not have any link to this company except an MTA, material transfer agreement, to get a free compound from this company to do research in my lab. That's all. I do not buy any stocks from this company.
I suppose all these supplements might be completely useless. I don't know if the nature of NAD is can be absorbed and can be really, I don't know. Well,
yeah, so I can very confidently answer this question to you. As I mentioned, there has been more than 20 clinical trials in the USA, in the UK, in Birmingham University, and also in Oslo. My head of department, who is leading a project on NAD+. So when we're talking about NAD+, we do not feed in the people with NAD+. We are giving the people NAD+ precursors, such as nicotinamide, riboside, or nicotinamide, which is vitamin B3, or nicotinamide mononucleotide. Speaking of the clinical safety dose of nicotinamide riboside, study has been shown 0.5 gram, 1 gram, to up to 1.5 gram per day. It means divided to 2. For up to 3 months, there's no detectable side effects, while it can increase blood and urine NAD+, as well as brain NAD+, using the MI approach by a University of Bergen colleague. Definitely, there's still a lot of debate because NR and NMN, when we orally consume those, majority of them can be degraded by our microbiota. So it is nicotinamide or vitamin B3 goes to the blood to be supplemented. However, studies from my group and others to show NR and NMN works better than nicotinamide. So there must be something we do not know can benefit the brain.
Okay, thank you very much. Congratulations to both of you again.
So many thanks to our two speakers for their interesting and very impressive talks, and many thanks to all the participants for this really lively discussion. Many thanks, and I wish you all a nice day. Bye.
Thank you, Kostas and Panos. See you guys next time, very soon. Yeah.